Hello and welcome to the I Can Do podcast with Benjamin Lee. We're here to talk about tips and strategies to have an I Can Do mindset. Life is what you put into it. Get the most you can. Here's your host, Benjamin Lee.
Hello and welcome to a new episode of the I Can Do podcast. I'm Benjamin Lee, your host, and so thankful that you are tuning in to another episode. Wherever you may be in the world, I always appreciate your commitment and your time listening to this podcast. There are so many different ones that are out there. As you know, if you've been following the I Can Do podcast, we've had a variety of guests, fantastic guests on the show. And today we have another fantastic guest, a friend of mine, a brother in Christ, and someone that will certainly benefit you and help you in your faith with God today. And maybe you're searching for the truth. Maybe you have questions about creation and evolution, and does God really exist? I believe what we're going to talk about today is going to benefit you. And so with that, I want to introduce our guest, and I'm going to start off by reading his bio. Today we have Dr. H.E. Buddy Payne with us. And let me just give you a few details. I have a brief bio that he gave me. Dr. Payne received his formal education at Florida College, an AA, in 1965. He attended Florida State University, where he received his Bachelor's of Science in Chemistry Education in 1967. Harvard University, where he received an MAT in Science Education in 1968. And the University of South Florida, where he received a PhD in mathematics education in 1983. He joined the Florida College faculty in 1972, where he taught chemistry and mathematics, served as registrar, dean of students, and vice president and academic dean. In 2009, the board of directors selected Dr. Payne as the 5th president of Florida College. He served in that role until his retirement in June 2022. Upon his retirement, the board appointed him as chancellor of the college, a role in which he serves till the present time. Brother Payne has served as an evangelist, teaching and preaching the gospel for churches in St. Petersburg, Cortez, Tampa, and Lutz, Florida. I don't know if I got that right or not. He has conducted Lutz. I got it wrong. I practiced it as well. He's conducted numerous gospel meetings for churches around the United States and has done foreign evangelistic work in Romania. He currently serves as an elder for the Livingston Avenue Church of Christ in Florida. And he has devoted much time to studying and lecturing on the creation evolution controversy on college campuses and in other settings over the last 60 years. Dr. Payne, welcome to the show.
A pleasure to be here, Ben.
I really appreciate you. Yeah, I appreciate you accepting the invitation. I reached out to you a couple of months ago, and we were just talking before we recorded just the interactions that we've had. We first met in Beaumont, and I know you were born and raised there. I was preaching at the Dowling Road Church of Christ, and we had started some studies on the Lamar University campus there, and I believe you gave 2 presentations on campus talking about creation and evolution, and I think a total of 500 people came out, college students, church members, professors, which was just really fantastic. And now I'm in Louisville, Texas, and you came up here last year to conduct our VBS, and this is what really got my attention even more with what we were talking about with creation and evolution, and in particular DNA, something that I really want us to dive into. So I just want to say thank you for your work and for your commitment to teaching and preaching the truth. I really appreciate it, and I know you've changed so many lives as well.
Well, it's been a joy, and especially here in these latter years, to be able to spend extra time on the topic of evidences for God in the natural world, because it seems to me God has opened the floodgates of evidence for his existence from the things we can observe. So that's what I want to talk about tonight.
Yeah, absolutely. So we had dinner at at a restaurant here in Louisville last year, and I had asked you, as a preacher and just even thinking about churches, what do we need to be aware of? What do we need to be concerned about, or at least, getting even more prepared? And I believe you mentioned the rise of atheism. There's a whole new section or group of people. They may not describe themselves this way, but there's a term, the nuns, NONES, that really have no religious affiliation. And we know atheism has been around for a long time. What really prompted you to get into, this path of just talking more about creation and evolution? Was it something that you experienced personally? Was it just you seeing what was happening on college campuses? Give us some feedback.
Yes, in my high school and early college days, I was a math science person. I really enjoyed and was passionate about mathematics and the sciences and chemistry in particular. So I sat at the feet of some very intelligent people, both at the high school and college level, who were strong believers that there wasn't a God and we didn't need God, and that so the sciences could answer a lot of the questions that religious folks answer. So I had some personal struggles with that myself and didn't know how to respond to some of the things I was being told. And the 50s and 60s was an exciting time for the development of the understanding of life at the chemical level, especially. And we'll talk about that some tonight. So as those things were unfolding, it seemed to me that it was a time when people were getting pretty confident of themselves, that man was going to figure out how all of this happened without God. And so that was a stimulation for me. I wanted to find out myself with an investigation and really openly figuring and looking, thinking and reading and studying and listening. And so it just became a passion of my life. And when I was privileged to get a scholarship to go to Harvard, I didn't turn that down specifically because I felt like that would give me an opportunity to dig in the sciences and sit at the feet of some of the world-renowned professors and dig even deeper. Plus, it would open doors for me going forward, because if I have a Harvard degree, that doesn't hurt anything. Talk about this topic.
Absolutely.
I'm not a scientist in the sense that I haven't practiced science myself and done laboratory experiments and, can come up with theories of things we didn't know before, but I am a teacher. All of my degrees are in education, if you noticed. I feel from the beginning, that's the passion God gave me, to teach people. And I think I do have the capacity to take difficult topics and explain them to people. So I taught math and chemistry for years and dearly loved it, because it was a delight to bring people's eyes to light up and understand something they didn't grasp before. So all that together, and that's my motivation. And to that, now that in our time, there's been a strong move in the last 20 or 30 years among scientists who are of a worldview that atheism is the proper answer. And so much so that they're almost evangelistic in that conviction. And their efforts have had an impact on young people. And I think there have been many young people whose direction in life has changed, even if they were brought up in religious homes. Many of them have lost their faith in God. Think that the answers are in science. And I think they're sadly mistaken. But I think they need help, which is why I feel privileged to have a part in doing some of this and helping young people see that there are answers to the criticisms that are made and to some of the arguments being presented.
Two questions just listening to you. We're going to do something unique on this episode where for the first time, I have a guest that's going to be using some PowerPoint slides. So if you're watching this via Spotify or YouTube or on some other platform, you'll be able to follow along with audio and slides as well. When you were going through this journey for yourself, Dr. Payne, was there a particular argument that really, I don't know if solidified is the right term, but reinforced okay, yeah, this is the truth. Like, was it an argument for design? Was it something else that stood out to you as you began reading more and studying more and really looking at the science and seeing what the evidence has to say?
For me, the argument from design has always been the strongest argument for the existence of God from evidences in the natural world. And of course, it was a very powerful argument in the 17 and 1800s.
Mm.
It's commonly taught that nature, natural theology was what the courses were called even in college in those days. And that where you were studying nature and you were showing how nature testified to the existence of a grand designer because there was so much design everywhere.
Is that the reason why that argument seems to be the most impactful, just because of everything around us?
I think it's because people who were looking in the days when science as we know it was unfolding in the 16 and 17 and 1800s, the people who did the best work in the sciences, most all of them were believers in God. And their view about that was, we're just thinking God's thoughts after him.
Mm.
And we're unfolding in nature the testimony of God's existence and what he has done to make the world the way it is. That was the common perception. Men like Copernicus and Galileo and Johann Kepler and Isaac Newton, some of the greatest thinkers of all time, and certainly helped in the development of modern day science, and lots of others that we can name, took the view that we are uncovering the secrets of God's creation.
Mm-hmm.
And we're really seeing how he himself demonstrates his presence in the natural world. That started changing in the 1800s in our modern time, because various ones began to say, no, we don't need God for this, we don't need God for that, we don't need God for another aspect of the sciences. And I would say for 150 years after Darwin's treatise in 1859, the turn in the academic community was toward not believing in God and toward scientific materialism. That has been the fundamental approach and still is in many places.
Mm-hmm.
So I believe what's happened is now in this latter time, we'll say the last 50 to 100 years, science has uncovered many things that we didn't know before that point more powerfully than ever
Mm.
to the designer. So if you want to show the first slide that we use, I like the design of this slide. It was done for me. I don't do this slide design very well myself.
Mm-hmm.
But I really like that slide because it says the argument for design revisited.
Mm.
And you see the picture of something that looks kind of like the DNA structure leading up into heaven. I really like that.
It looks great. It looks great.
At any rate, that's the thing I believe I'm trying to do
Okay.
as I talk to people about the things we'll discuss tonight and lots of others, because I truly believe we have now a grand opening of many things
Mm-hmm.
that help us see more clearly than ever. There's powerful evidence for God in the natural world.
Absolutely. And just listening to you speak here, and we'll dive into the slides here. I'm always curious, what's the response when you have presented this kind of evidence at different colleges? What kind of feedback do you get?
Well, of course it's varied. I don't know how much time you want to spend on this, but I have two events in my life that I think are the highlights in that sense.
Okay.
So asked to do this kind of a series on the campus of the University of Maine many years ago.
Mm.
And there's a little church there that was involved that helped to sponsor it. And they probably had 25 people or 30 in that church, members. So it was a very small group, but they rented a room on the campus of the University of Maine.
Mm.
And it was a science lecture hall, which is where I love to do this, because I like to be right in the middle of the science setting, because we're not looking at the Bible in the series that I do. We're looking at evidence from the natural world.
And why do you make that distinction? I think that's an important distinction. Obviously, we're not saying the Bible is not important, but you do make that distinction. Why is that the case?
Make that distinction because if you're talking to people who are not believers in God at all, to quote from Genesis 1 and say that in the beginning God created the heavens and the earth, and there's a statement from God himself saying that he created, that's not the scientific approach to the answer to this question. Science doesn't take a textbook that claims to be God's revelation and start with that.
Gotcha.
Science starts with examining the natural world and coming up with what they believe is the most reasonable conclusion as to how that came to be. So when I talk about this subject, I want to talk about what is science saying to us from the observations of the natural world that we are capable of making now with the instruments we have to assist us. And to me, that is the proper approach when you're going to try to talk to someone about why they should believe in God when they don't even believe in him at all.
Yeah. I think that's good. I think that's something important to emphasize. So let's talk real quickly, and then I'll move the slides for you. And for those who are listening or watching,
Well, let me finish my little story there about the University of Maine.
Okay.
We found out later because when we went to our first night for that lecture, I did 2 lectures each night with questions and answers in between. When we showed up, there were about 15 of us who were coming, who were in the group that sponsored the event. There were over 200 people in that room.
Wow.
We found out that the president of the University of Maine at that time was a member of the Mormon Church. And he saw the advertisement on campus and liked the idea, and he asked his faculty to go to the lecture and take their students. So I came into the room that evening. We had, I'd say 95% of the people in that room were not believers. And their professors were there. We had biology professors, chemistry professors, physics. We had people from the philosophy department. There was a lot of faculty there, in addition to a host of students who were there. I think they had heard this guy from Harvard was going to come and talk in favor of creation. I think they came to see a dinosaur. They didn't believe that there could be somebody there who was a Harvard graduate who still believed in God.
Yeah.
So that's why we had a crowd, I think.
Yeah.
They really believed, I think, that I was going to open the Bible up and let the Bible be the textbook for that discussion.
Gotcha.
Well, I didn't use the Bible at all the whole time.
Yeah.
We strictly looked at the evidence of the natural world and showed that the most reasonable explanation, at least in my mind, for that is that there must have been a grand designer who had grand intelligence. It could not have come about in any reasonable way otherwise.
Yeah.
So that was one. And at the end of that series, I got into it with a Dr. Cornfield, who's the biology teacher. I thought that was a good name for a biology teacher. And we joked with each other a lot because I'm Dr. Payne.
Yeah.
I've been called the Payne and everything you can imagine. But I always tell him my name is spelled with a Y.
Yeah.
It's the hurt out of it.
That's great.
So we did a lot of joking about names, but he and I were back and forth a lot during the Q&A.
Yeah.
And by the time we finished that weekend, he and the head of the science club on campus were inviting me to come back.
Really?
So I really thought that was a success story.
Absolutely. Now, you said you had another story. We got time. What's the other story?
Yeah, the other one was in California, where a church had asked me to come and do a series like this. And I told them I much prefer to speak in a neutral setting. So they had rented a huge facility. It's near Los Angeles. It probably seated 500 to 600 people. And that was not the whole building. They only used a part of it.
Mm-hmm.
We probably had 500 people there for the lectures. And I think that's one where I really grew up in my presentation, because sitting on the front row of all those people was a row of evolutionists with their
Really?
computers checking every word I said.
Wow.
And we had Q&A. And so they were able to open up their computers and check what I was saying and challenge me on just about everything. So it was a grand experience to have that kind of a setting and have that many people there.
Absolutely.
And then have people who were not believers listening and challenging and going back and forth. And I think the great majority of the people there would have said that was a successful experience, because the evidence for design was clear.
Yeah. Yeah.
At any rate,
I'm curious before we move forward. You got to tell me in the audience, how much time did that take for you to prep, right? There's a lot of pressure to
yes.
go into a college campus. You have no idea who's the audience. Can you give me some feedback with that? How long did you prep leading up to that to be ready?
Well, I would say that happened probably, I'm not remembering the year exactly, but I think I had been doing this kind of thing for about 10 years by that time.
Okay.
So I would say 10 years. Because
So,
really, this had been a passion of mine, that every spare minute, my wife could tell you that when I was at Harvard, every spare minute was in Widener Library looking for everything I could find by anybody on this general theme. And I can tell you in those times, there was not much there written by believers in God that was worth reading.
mm,
There was hardly anything at all on this general topic. And now there's a lot of wonderful material written by credible scientists
mhm.
who are strong believers in God because of the evidence of the natural world. That was not the case in the 60s.
Yeah. Yeah.
All of that, you ask for my own personal motivation. That's where I've been motivated. And I think what I've done has helped many people see something they hadn't seen before.
Amen. Yeah, absolutely. Yeah, that I'm always interested in that, and I think there's something to be said there about this passion and how it's still even needed today. And that's one of the reasons why I wanted to do this podcast, and hopefully even more podcasts. What I to the audience, I had talked to Dr. Payne, and I said, I think it'd be good for Dr. Payne to have his own podcast. You just have so much knowledge, and it's just such a great opportunity. But let's dive into this. And what we're going to do here, this is still going to be conversational. So we want to talk about the argument for design revisited, and then arguments against design that I guess you've heard and people sometimes throw out, and how we can navigate this, and at least just understanding what these arguments are, and then pointing back to the science. What is the science actually saying?
That's a good thing to say. So what we're going to do is use some slides to help with visuals, and we'll be skipping around some. But the reason I want to go this direction is because it does two things. I want to talk first about the argument for design as it relates to the chemistry of living things.
Okay.
So we're going to talk about DNA and proteins and the things that make up living things. But then we're also going to talk about junk DNA, as this came to be called in the 90s and the early 2000s, as we began uncovering more information, and why that was a challenge to believers in God, and how that's turned out now, some 10 to 15 years, 20 years later.
Yeah.
I think it's a good illustration of how science can be both a challenge and a grand support to belief in an intelligent designer.
Yeah. Awesome. I got to ask, what is this photo on this slide here?
Yeah, well, that's an anomaly, because I did this lecture first when I spoke about the poor design argument in general.
Gotcha.
And we talked about the human eye as well as junk DNA. And some argue that the human eye is not a good example of how there's a creator who designed it, because the design of the human eye has lots of problems.
Mm.
That's one of the arguments made. by evolutionists and atheists. So I'm responding to that. Well, this particular picture is a picture of a type of it's a cell that crosses over your eye, part of your eye. So
Gotcha.
it's not our topic tonight.
Yeah.
It's an illustration of how something that has come to light for us in more recent days undergirds design argument.
Gotcha. All right.
It's about.
Okay. So I'm going to move through these slides for those who are listening and those who are watching as well. And I think what we can do, I'll just put up the slide and we can move around as you like. But you want us to start here. I know you don't really use the Bible that much when you go to these certain presentations on campuses because you want to emphasize, let's go back to the science. But walk us through the thought here on these verses and what you want to get across.
Well, when I talk to an audience that has a mixed crowd in it, Christians and non-Christians, I like to start with this because I want to say that the Bible does proclaim in that book that there's evidence for God in the natural world. So Psalms 19 in the Old Testament, Romans 1 in the New Testament, both testify that there is evidence for God. The heavens declare the glory of God. And there are invisible attributes of God that can be seen, even though he's invisible, by the things that are made. So the argument is, if you examine the natural world and you see things that are clearly designed, it leads you to believe there was a grand designer.
Yeah.
And that's the very thing said in the scriptures as well.
Yeah.
So that's the argument in its simplest form. And that's the argument that I make over and over again from all of these various levels of scientific investigation. So, for example, the cell. Darwin, in a way, may be excused for this because he thought the cell was some kind of a little glob like jello. We knew almost nothing about the inner workings of the cell in the 1800s. And he wrote his book, of course, The Origin of the Species, in 1859. And at that time, the things you see on the screen right now, all the different parts of the cell, he knew nothing about any of that. He thought it was very likely that a cell could just arise naturally because it was so simple. So, as I said, you could almost excuse him in some ways for that, though he really didn't have much to say about the origin of life itself. Right. His theme was how do living things develop after there is a living thing. He did make a comment or two about how maybe out of some warm little pond there might have risen the original life form, but he didn't make a thing out of that, and certainly not in the origin of the species. So I would say one of his points that he makes in his book, The Origin of the Species, is if you could find a complex organ that existed that could not possibly have been formed by slight, small, sequential changes over long periods of time, my theory would absolutely break down. His theory is even complex things can come about naturally by small scale changes adding up over long periods of time. So now the thing looks like it was designed, but it wasn't. It was just small changes adding up over time that happened naturally. And the small changes were small enough that they could have come about by natural causes. And today we would say, he didn't know much about this piece of it, but today we would say mutations acted upon by natural selection can bring about changes that eventually produce a new species. And over time, all the species that exist could have come about that way. So that's the simplistic explanation of what he said. He did not know the complexity of the cell, and I would present this as a classic case for him of a complex system that could not possibly have been formed by small successive changes over millions of years.
So he was lacking very important evidence that we now have in science at our disposal.
At least on the cellular level, yes. But he made a lot of observations now on living things, and he's to be credited for being a great discoverer of lots of properties of living things and organizing that information.
Yeah.
He didn't understand the complexity of the chemistry of living things by any means. And that's what we've unfolded in the last 50 years that, or 100 years, that he would not have known about at all. So inside this cell, you see the nucleus there in the purple. We know now that most of the DNA in a cell is in the nucleus, not all of it. Some of it is in mitochondrial DNA, and there's some other things, but the great majority of the DNA in a living being is inside the nucleus of a cell in eukaryotes. In prokaryotes, it's more spread out in the cell. But the DNA is inside the cell. And that DNA is in charge, really. It's directing the processes that take place. Well, DNA was not really discovered and really investigated till after 1950s. So that's your answer. That's a long answer to what you said.
That's a great answer. That's fantastic.
And I would argue that every living cell is full of signs of intelligent design. Everywhere you look, it is a massively designed thing. And so many of its features, they point to somebody designed this. This did not come about by natural causes.
What's some of the data people, not data, but some of the examples people say, like in DNA, with the amount of information, equivalent to, what is it, 1,000 or thousands of encyclopedias. Have you heard of different things like that?
Oh, yeah. One of the great mysteries for an evolutionist, an atheist, is how did that much information get crammed into the DNA?
Right.
And one of the fundamental arguments for design is, in all of our experiences as humans, and we're the only ones that are sitting around thinking about this, by the way. We don't know of any
that's right.
living being that's thinking about all of these things.
That's right.
In all of our experiences as humans, we have zero evidence that information comes about apart from a mind.
Yep.
It is a result of an intelligent mind. In all the things we've observed where we can know it for sure, lots of things have been made by intelligent minds. We know we've done it. But the fact is, something like a language that contains information and can communicate information, that came from a mind. That didn't come about by natural causes.
Mm-hmm.
And so did hundreds and thousands of other things.
Yeah.
It's a natural thing when you see evidence of information to think somebody designed that. Well, Crick and Watson are credited with discovering the structure of DNA in the 1950s, for which they won the Nobel Prize later. Mm-hmm. I might say that they made kind of the last step in that when a lot of folks had preceded them. They stood on the shoulders of giants in their discoveries, but they got credit for actually uncovering the structure, which the next slide will show you. And go ahead and click on the words that are coming up. It was a double helix with crossbars, as you can see over in the picture. And there were 4 bases that made those cross pieces. And so they're listed here: thymine, cytosine, adenine, and guanine. And I've color-coded them to fit with the colors on that double helix over there. And you can see then that the red always joins up with the yellow, and the blue always joins up with the green. And that's because of their chemical properties. So when you look at that strand up and down, and imagine unzipping that thing, taking it apart, you expose the 2 sides, and on the 2 sides, you have a sequence of those 4 chemicals. And those then become the alphabet for DNA. Gotcha. And what we've learned is not only did they allow this process, and this structure allows it to reproduce itself, it also has contained in it a language. that we've learned how to read and we know what it means.
Mm.
So here you have in every living cell a functional language that is doing things, in particular, building proteins at an enormous rate every second. So it's a language that's being transcribed, translated, and used to build things. And a whole lot of other things we've learned out in recent years. Well, that in itself would make a great case if you had a jury, you were trying to convince them there's a God. If you have things in every cell that has a language in it, that's a sign that somebody intelligent did that. It didn't just come about by natural causes. So, but let's go on.
Well, two thoughts too. One thought is we always have to be willing to follow the evidence. And I think that's what a lot of people who do talk about science, be willing to follow the evidence. So as this science, new science and information emerges,
Yes,
we have to be willing to not double down on something, that we thought when we have new evidence presenting itself, for sure. I think that's something important too.
no question. So scientists found then over time that not only was the structure amazing and allowed for it to reproduce itself, but it also contained a language, and we learned what the language meant and how to read it. And it was a description then of the process of building proteins. That's how it unfolded first.
What's the significance of us being able to not just decode, but also to read the language?
Well, what it means is it's a language that makes sense. It has a meaning that has some purpose.
Gotcha.
And so here you have a language that has information in it, not just any sequence of those letters does something. Some of it's just gibberish if you have a particular sequence. There's certain sequences that do things.
Gotcha.
And so I guess we need to go to the next slide, because here's a picture of what happens with that DNA. You see up there at the top, it says transcription. That's the DNA across the top up there. And it contains the information, but the information has to be transcribed, copied. So can you see where that DNA has opened up there? It's unzipped.
Yep.
When you unzip it, you've exposed the two sides. And what the body does, what the cell does, is it copies the information onto what's called messenger RNA, which then carries it somewhere else and does something with it. I'll describe in a moment. But the first thing you have to do is copy the information out of the DNA, and then you have to transfer it in order for it to do its work. So the first step in this process is transcription. Well, that happens up there. The DNA unzips, the information is copied, and then it's carried outside the nucleus and it's acted upon. So there's transcription, then there's migration as it moves, and then there's translation. You see down, it looks like two ducks hugging each other down here.
Yeah.
That's a picture of ribosomes, which is another organelle in that cell. And it picks up the messenger RNA. You see that tape coming through there? That contains the information that is on that DNA up top. But it has to read that information, and then it uses it to produce the protein you see coming out the bottom there. Well, the protein is a sequence of amino acids. But the amino acids are coded in the DNA. There's a code for each of the 20 amino acids in all common proteins. And so the code is read, protein is then collected together, and the amino acids are combined into a string, and you have a protein. So transcription, migration, translation, and you end up with a protein.
You can't help but think about design. I mean, the chances of all
the plan to me.
Yeah, it looks like a plan. There's organization, there's structure, there's order.
That's right.
And the idea of all of this happening by chance.
Well, here's
Yeah.
a big piece you need to understand, and this is
Okay.
what I'm trying to get folks to see. In order for you to transcribe the information off of the DNA, you need proteins to make that happen.
Hmm.
But
So
what are you trying to do with the process? You're trying to build proteins.
yeah.
Well, the proteins you need up there in step one have to also be built. So
It can't be this slow.
so how did you get it in the first place? That's right.
Exactly right. Yeah.
You need proteins to make you be able to build proteins. In fact, in this whole process, there are at least 50 different proteins required for the process to be completed. It starts up at the top there. There are proteins needed to make that thing unzip and allow it to be copied. There are proteins involved in the migration. And do you see there that right below the DNA up there, there's a little strip of messenger RNA?
Mm.
You see it has some white things in it?
Hmm.
You see those little white places?
Yeah.
Those have come to be called introns.
Mm.
And you see that the next thing of that is kind of snip those out.
Mm.
That's been edited. It's called matured. So the white things have been snipped out or rearranged. So you have a consistent sequence of exons, they're called. Those are the things that code for the protein. So we've snipped out the things in there that don't code for the protein and put it back together. That's called maturing. That requires proteins to do that.
Yeah.
All right. So once that's done, when you migrate through that cellular wall there, the nuclear wall, it takes proteins to get in and out of that wall. You have to open up and allow it through.
Mm.
There are also proteins involved with reading and gathering up the appropriate amino acids out of your cell. And then to put them together and make a string of amino acids, which is a protein, also requires protein.
Mm.
So think about this. We have discovered that in every living cell that's a eukaryote,
Okay.
there's information in the DNA that has a language in it. There's a process by which that language is copied, migrated, and then used to translate and make proteins. And every step of that process requires proteins, which have to be built by the same method. Sounds like that all came about so that it could all happen, not that it evolved over millions of years, step by step, small sequential steps.
Mm-hmm.
No, this points to design, intelligent engineer.
Yep. Yep. That's exactly right. I mean, it's so powerful just to be able to listen to this and to see it. And
well, here's what scientists have done with that. You have the next slide up, which has blown up that one piece, right?
Mm-hmm.
You see where those little introns, those little white things have been snipped out?
Mm-hmm.
The argument began to be made in the 1970s and 80s and 90s that it's pretty clear that not all of that DNA is used to build proteins because these pieces are snipped out.
Mm.
And it began to be thought, well, if they're snipped out and removed, then they're not needed. And why would some God make something where you had to snip it out and edit it? Why not just make it so it's got everything you need right there and you can move right on? That looks like the evidence of an evolutionary process that didn't turn out exactly the way you wanted and you had to fix it.
That's a great question.
Yeah. So the introns were thought to have no particular purpose except they're in the way.
Mm.
So you snip them and get rid of them. And it's just garbage. And the thought was, what you've got there is a piece of junk left over from your evolutionary past that didn't work out right. It certainly wasn't something that sure looks like it was planned perfectly well. It looks like it's a leftover. Okay? That started coming to light in the 70s and 80s. And there was a gentleman forget where he was from. I think he's from the Far East, but he made this statement: why does the DNA have so much junk in it? That was the first one that really made a statement like that. He was a
Junk DNA. Yeah.
Yeah. And so the more they studied and learned about what the DNA was like, it became more and more obvious, they thought, that there's a whole lot of junk in there left over from what clearly looks like a hodgepodge method that would speak to evolution, not creation.
Gotcha.
So DNA, which was used by creationists for a good long while as grand evidence for a God, now is being turned into a terrible evidence. It's a poor example of design, you see?
Did that ever stump you, or did that ever cause you to pause?
It was very dramatic for me because I'll tell you, when I first ran across this, I was doing a series of lectures on the campus of the University of South Florida, and we probably had 100, 150 people in the audience, many of them not believers, and I was presenting this kind of material on the DNA. And I had a professor stand up during the Q&A and say to me, Professor Payne, you did a nice job of explaining the amazing process of protein building. And it's utterly amazing. Truly is. But what you didn't tell the class is that that only takes about 5% of the total DNA in your body. And we've learned that the other 95% is mostly a bunch of junk. Why didn't you tell the class that?
Oh, no.
Well, at that point, I had not studied that very well, was not well informed at all. And I told him, I don't have a good answer for you. I said, because a believer in God would certainly not think that God would make something 95% junk.
Right.
That'd be hard to explain, wouldn't it, from a creationist standpoint?
Mm-hmm.
I said, I will certainly be studying more about that. But I said, I do want to say one thing to you. I don't have a good answer for you tonight. I'll be looking at this very carefully. I said, but I do remember from my study of history of the theory of evolution that there were scientists in the 18 and early 1900s who said your physical body was a veritable leftover of your evolutionary past. And at one point, they were saying there were at least 150 vestigial organs in your body. Like, you know any of them? We thought of as a vestigial organ, something you don't need. It's a leftover.
Appendix?
Your appendix is a classic.
Mm-hmm.
The muscles of your outer
ear, yep.
Your tailbone, left over from when you were a monkey.
Mm-hmm.
Those kinds of things. But there was a list of about 150 of those. You were a veritable, like, leftover of your evolutionary past.
Mm-hmm.
Over time, as we learn more about the way our bodies function, practically all of those 150, nobody says are vestigial anymore.
Yep.
When I was a young man, my adenoids and my tonsils were taken out. They were considered vestigial. And a lot of kids had those removed. Well, we've learned since that you shouldn't be taking those out. It's best for the human body.
Right.
So my point to him was, we've been down the road of calling things vestigial or junk left over from the evolutionary past, and we've been embarrassed by it. I said, maybe the better thing to say is we don't know what's going on with that other 95% yet.
That's a great explanation, yeah.
Instead of calling it junk.
Mm-hmm.
That's not what they did. They called it junk. So I have some quotes here. Let's go on and let me show you some quotes about what was being said.
Okay.
This is a little summary of what I've just told you. What about the rest of the DNA? Some again describe the remaining parts as junk.
Mm-hmm.
And when the Human Genome Project, which took place from 1990 to 2003, do you know what that is?
Yeah, I remember it was a really big project. One thing that just comes to my mind when I was looking at that, I think there was some, I think people were actually looking if there was like a gay gene. They were looking for a lot of different things, but I can't remember every single thing about the Human Genome Project. But I believe that was one of the findings. There is no gay gene, but I don't remember all the details of it.
Well, the purpose of that project was to map the entire genome for a human. In other words, let's make a map of it. It's made up of these sequences of bases. I told you, you remember? There's thousands of them in our genome. So the goal was to map that entire thing. And it took them 13 years with people from all over the world helping. And it was guided by Dr. Francis Collins. You may have heard that name. Ended up being the head of the NIH. A believer in God, by the way.
Oh, I didn't know that.
But they found out a lot of things about our DNA, our human genome, from this project. And some of those things kind of supported the junk DNA theory.
Oh.
So here's a quote. Kenneth Miller, college biology professor. The human genome is littered with pseudogenes. A pseudogene, if I may interrupt the quote, is a gene that doesn't work anymore.
Gotcha.
It looks like it was a functioning gene at one time, but now it doesn't work. The human genome is littered with pseudogenes, gene fragments, orphaned genes, junk DNA, and so many repeated copies of pointless DNA sequences that it cannot be attributed to anything that resembles intelligent design. You see?
Yep.
Go to the next one. Here's Richard Dawkins, our favorite professor who is a strong atheist and has written lots of books about why you have no business believing in God. In fact, he says you're deluded if you do.
Yeah.
God Delusion is one of his great books that he wrote. It was a great, I think, #1 seller. But in his book, The Greatest Show on Earth, Evidence for Evolution, he said, it is a remarkable fact that the greater part, 95% in the case of humans, of the genome might as well not be there for all the difference it makes.
So you got a trend going on here,
I should say. And this man is the professor of he's to give public an education on the sciences. That's what they appointed him to be.
Wow.
So when
he's got a ton of influence. That's what you're saying.
He
then.
does. I mean, he's speaking for Oxford University and telling the humans who are not into the sciences what they ought to believe about the sciences. Well, he speaks with some authority. So here's another piece of that quote. Pseudogenes are genes that once did something useful, but have now been sidelined and are never transcribed or translated. What pseudogenes are useful for is embarrassing creationists.
He's mocking the creationists.
Yeah, of course. It stretches even their creative ingenuity to make up a convincing reason why an intelligent designer should have created a pseudogene, unless he was deliberately setting out to fool us.
Wow. And this was in 2009 when he said this in the book.
Common statements being made at that time. And I'm quoting to you from a book called The Myth of Junk DNA by Jonathan Wells, which came out in 2011, in which a lot of this has been debunked. But it hadn't been right away. And so I was going to read you a little more of that statement.
Yes, please.
Richard Dawkins in this book. And what I need to do is look and get in my notes and get the page number. So hang on just a minute here.
Yeah, go ahead.
All right, so this is page 26. All right, let's see. I think what I'll do is back up and read you a little more of Ken Miller's.
Okay.
Who was the one we quoted prior to his.
Okay.
And let me see where that one is. That's on page 22.
Okay.
It says, in fact, the genome resembles nothing so much as a hodgepodge of borrowed, copied, mutated, and discarded sequences and commands that had been cobbled together by millions of years of trial and error against the relentless test of survival. It works, and it works brilliantly, not because of intelligent design, but because of the great blind power of natural selection to innovate, test, and discard what fails in favor of what succeeds.
Mm.
See? So the junk DNA argument is saying the poor design of DNA is an example that there is no God.
There is no God. There is no designer.
And evolution did it, and that's why you got such a mess.
Right.
And believe me, that was said over and over and over again in the 1990s and the 2000s, early 2000s, until we began to uncover what that other 95% is doing.
Mm.
So after the Genome Project that mapped the whole human genome, it certainly did not answer 10,000 questions about what's going on in there.
Right.
So a new thing was started. That's your next slide.
Okay.
It's called ENCODE,
Mm.
the Encyclopedia of DNA Elements. Its goal was to identify all the functional elements in the human genome.
You have this at 2003 to 2012. That would have been right after the Human Genome Project.
Correct. So this project was entered into by thousands of scientists around the world. It was a collective process.
This would have been scientists who may have been creationists or atheists.
Absolutely.
Just a variety
Amen.
of scientists. Okay.
Yes. So let's look at their goals. The primary goal of the ENCODE project is to determine the role of the remaining component of the genome, much of which was traditionally regarded as junk. That is DNA that's not transcribed to build proteins. You understand what I'm saying?
Yeah, and I'm just thinking as you were reading that, I'm just thinking how quickly that term junk stuck. It got sticky, right? I mean, it's good marketing, you know, and so just their goal going back to 2003, how fast that actually became kind of like the norm.
Well, the guy that first mentioned that was back in the 70s. So that means
Oh,
it's been building up
it
over
was
time.
building up. Okay.
It kind of
Gotcha.
came to a crescendo after the Human Genome Project.
Gotcha.
So what these folks wanted to do was let's find out what's happening with the rest of that DNA that some people are saying is just junk.
Mm-hmm.
Second, it is to link variations in the expression of certain genes to the development of disease. Their thought was the more we learn about the human genome, the more we're going to understand how to help people who have diseases.
Mm-hmm.
And then thirdly, to build a comprehensive parts list of functional elements in the human genome, including elements that act at the protein and RNA levels, and regulatory elements that control that, and most circumstances in which a gene is active.
Mm.
So they're trying to discover what's the rest of that doing.
The rest of the story, the 95% that a lot of people are just dismissing as the natural result of evolution.
Correct.
Gotcha.
And leftovers. Or as Dawkins would say, you might as well not even have it for all the good it does.
Mm.
That's how he described it. All right, so what have the results been of ENCODE? Over 80% of the human genome is actively involved in at least one or more biochemical reactions associated with gene regulation of at least one type of cell. That was a total shock to them.
So
That over 80%.
yeah, so 95% is no longer junk.
No, not, we were saying only 5% is needed for making the proteins, and there are thousands of proteins that are built.
Mm-hmm.
But it appears only about 5%. In fact, it's less than that now that we believe are used for that purpose. But what they discovered is over 80% of that human genome is being used for something.
There's still purpose.
Amen.
And design, yeah.
And then here's another quote from the ENCODE results. The human genome may contain only about 21,000 genes, but the ENCODE scientists found 70,292 areas called gene promoters.
Wow.
That precede the protein coding areas of the genes.
Mm.
So let me explain that briefly. There are sections in front of the coded part that promote whether that's going to be used or not.
Wow. That's amazing.
What we've learned is it's part of a control system built in.
Gotcha.
You know, like with a computer, if you have software, you can put it on your computer all you want, but if there's not an operating system for your computer, it's not going to work. So the way to think about this is what we're learning is the rest of this DNA, a lot of what's going on here is a massive operating system for all that's taking place.
So you have code, you have language, operating systems,
Correct.
order, and we just keep finding more and more, which again goes back to what you were saying about Charles Darwin. He didn't have all the details. He didn't have all the science that we continue to
That's for
have.
sure. He
Mhm.
really did. Certainly not at the chemical level.
Yeah.
That's why, in my mind, this is one of the grand black boxes that God
Gotcha.
opened up to us
Mhm.
that Darwin knew nothing about. And by the way, there's a great book written called Darwin's Black Box by Michael Behe, who's a wonderful creationist, but he's a thorough scientist who does
Gotcha.
careful work. All right. Gene expression is controlled by a broad array. So what we're thinking is that the gene that is used to make the proteins is controlled by a whole bunch of processes going. So for example, there are regulatory proteins needed. There are chemical marks in the DNA. There are gene promoter features, which we just talked about, specific DNA sites. And then there are enhancer sequencers, sometimes located millions of bases from a gene or a set of genes. You catch that? If the gene is sitting right here, there may be an enhancer way down that DNA strand, a long way away from it, that has an impact on this and when it's how it's done.
Yeah.
So
Wow.
it illustrates. So here's the next picture. And of course, we're not going to be able to go into all of this, but this is a kind of a combined project picture to show you some of the results that have come out of ENCODE. Wish I had time to talk about each piece of this. But if you look at the green, you see the green down here?
Yep.
Those are the genes. You see the red piece? That's a little promoter area right in front of that gene. And then you see the yellow way down here to your left?
Yeah.
That's the enhancer-like elements that are a long way away from that on the DNA, but they do have an effect on it. We know that now. So all of that's been learned. And then you see all this up here at the top. That DNA has more information in it than we ever dreamed because it's all wrapped around itself and it's a massive amount of information. How do you get to that? It's like, how's your desk at home? You know, in your filing system.
Yeah, that's
The
right.
more stuff you get, the harder it is to keep it organized and
Keep
get
it organized.
Imagine the massive amount of information stored in here and how that system gets to it and works on it and uses it without everybody running into each other and all getting lost. I mean, it's massive.
I think that's another place to say too, right? You correct me if I'm wrong, is it more reasonable to think that this was all by accident over millions and millions of years, or that there had to be a mind behind it to fit all of this into the DNA and in the protein?
No question. Because let's go to the next one.
All right.
What about the pseudogenes? Well, we haven't answered all the questions about that. But some of those things they call pseudogenes have produced functional proteins. And some of them have produced RNAs that suppress the expression of their corresponding functional genes. So they're doing something. And some produce RNAs that increase the expression of their corresponding functional genes. All I'm saying is what were called pseudogenes may not be pseudo at all.
Gotcha. So if I'm hearing you correctly, then too, as we go along this path and this journey of DNA, the idea of junk DNA, which started in the early 70s, had this crescendo in the mid 90s and early 2000s. As we continue to open the black box and, look more at the data, junk DNA really is not going to be able to stand if it's standing at all, even though we may not have an exact answer of exactly what these like pseudogenes are doing. There's purpose and there's design in all of it.
That's what it's looking like. That's why Dr. Wells says we're now coming to the age of the myth of junk DNA. And frankly, most people have stopped using that term.
What about Dawkins? Has he retracted anything? That was said back in 2009, 2012.
As far as I know, he hasn't, but he has backed off on saying that argument as much, as far as what I've heard.
Is he still doing debates? I know he was, I
Well,
believe he used to do a lot of debates.
He has. I'm not sure. He's getting on up there like I am. He's 80 something. I'm 80. I'll be 80 this year. So,
Oh, okay. Yeah.
some of us are
Gotcha.
getting. All right, let's go on.
Okay.
This is the introns I was talking to you. Little white things, remember? They were
Yep.
snipped.
Mm-hmm.
What have we learned about those? Because those were called junk.
Well, we know that they're not junk. We know that they are playing a purpose.
Well, let's go to the next slide.
Okay.
Introns have functions. Those little things that are slipped out, they're rich in splicing factor recognition sites. They encode a majority of the small RNAs. Ben, we have learned that there are a massive number of RNAs we knew nothing about. There's small ones that come from the introns, a lot of them, and that have specific tasks that they carry out. And there's so much more to learn still.
Yeah.
But we know they're doing things, and they're very important. Don't tell me that's junk.
Yeah.
And then RNA from introns influence gene expressions by modifying chromatin. That's something else they do to help determine how a gene is being expressed. That does come.
Okay, I have a question. So we've had the Human Genome Project. We've had ENCODE. Is there any other big landmark project that's taking place currently?
Yes, and I don't have the name of it here in front of me, but yes, there's another one that's been going on. It's called Phantom. FANTON. It's functional annotation of the mammalian genome.
Gotcha.
There's other ones going on even still, and even the ENCODE project is being followed up with other projects.
Gotcha.
So let's go to the next one. Here's another just amazing thing that we learned through ENCODE. There are multiple messages stored in the same sequence of bases.
Wow.
Now, I want you to go back to that previous slide where we were looking at the introns. Can you go backwards?
Yeah.
There, this one. You see, when you snip those out, you get a mature one.
Mm-hmm.
But can you imagine that if you snip those out and you put them back together in a different order, you could make a different protein?
Wow.
You see?
Mm-hmm.
So what we've learned is that rather than being a leftover piece of junk from our past, this is a brilliant plan to allow the same piece of information to build 10 or 15, and in some cases, 1,000 different proteins.
So what we thought, or what some thought was junk, we could say God was laughing at them.
Amen.
And saying, if you actually only knew what I've created.
Yes. So look, first of all, go back to that. Don't leave that one yet.
Okay.
When you snip out those introns, guess what it takes to snip them out in the first place?
Protein?
Proteins. In fact, it takes spliceosomes, and I'll talk about that in a minute. And to put them back together takes, what do you think?
Protein.
More proteins and other things like RNAs that are called editosomes, where they put it back together again. But see, they don't necessarily just put it back together in the same order in which it came. They could change the order, and that's exactly what happens.
Wow.
So where you might have a 1, 2, 3, 4, you could have a 1, 4, 3, 2, or a 1, 2. You could change the order of the sequence. That's a different protein. So the same section of information, by this complex process, can generate for you many different proteins from the same piece of information.
Wow.
So DNA has in it way more information than we ever dreamed because of this process. And the things they thought were junk are vitally important pieces, not only for that, but for other reasons that they're doing while that other's going on.
More than meets the eye. I think, I can't help it. I know we're looking at the science, but Genesis chapter 1, everything was good.
Yeah. Let's go back to that slide we had a minute ago about the
okay.
Keep going. That one. Back.
Okay, there we are.
Multiple messages stored in the same sequence of bases. I just described that. And you need spliceosomes and editosomes to make that happen. And that's what I want to talk to you about next. So next slide.
Okay.
What's a spliceosome? We'll take a look at it. It's the 4 bluish blobs up there at the top. You see the strand of DNA copy, that's the messenger RNA sitting there. Got exon 1 on the left and exon 2 on the right. And then there's a strand in between that's the intron that's going to be snipped out. The blue things together make the spliceosome. So let's read it. Okay. Several different snRNPs join with other proteins to form a spliceosome. A spliceosome is an extremely complex thing that's made up of different kinds of proteins that together snip that thing out, get rid of the intron, and put the other back together. It interacts with the RNA, cutting out the introns and splicing together the exons. And if we had the time tonight, we could show you a little, at the bottom of that slide, it says splicing animation. See that? So we could go and show you that animation of this thing doing the job. We know exactly what it does now.
Yeah.
That's another complex system that's working all the time, and it allows a whole bunch more flexibility and immense design.
Amazing. Yeah.
A spliceosome is an incredible motor machine. Ben, inside every cell, there are thousands of machines doing all kinds of amazing things we never knew one thing about 30 years ago. And they're working in there doing amazing things that are way more complex than we ever dreamed.
So with all of this, someone who may be listening, or someone that we may actually talk to at some point in time, whose mind may already be made up with respect to Darwin and evolution, we can take away from this. You need to take another look.
Open your eyes again, please.
You need to open your eyes and open your eyes with the science. And can we also say this too, buddy? I know we're going to move on.
Yeah.
That science has never really been the issue, but it's been how we interpret the science, or maybe our own biases as we look into the science. Is that correct?
I believe that. Well, if you start with the notion that science only allows materialistic explanations, in other words, science does not allow God's foot in the door. And I can read you quotes from a Harvard scientist who said exactly that. His name is Lewontin. We will not let God's foot in the door in science. In other words, you're not going to allow an explanation for some intelligent being that isn't a material thing. That's out. So if you exclude the only other possible answer,
right,
which is the only other possible answer,
right,
then you have the answer. You're going to find some way to explain that naturally.
Right. Dr. Payne, one more before we move on. Can we also say then that the conflict that a lot of people have made between God and science maybe is not as much of a conflict if we take away our own prejudice or bias or assumptions as we look into the science?
I think that's correct. And to be fair, I think it's the honest man would say people on both sides of this question have made foolish statements.
I was going to say that some Christians
have overstated their case,
and they're afraid of science.
Correct.
Know
that.
Yeah.
So I hope we can find a middle ground here where we're allowing an explanation for intelligent design. Look, science cannot investigate a being that's not material.
Right.
It cannot. So I understand that problem. Does that mean it cannot point us to evidence that sure points to a designer? No, it does not rule that out. If you want to learn more about that designer, you may have to go somewhere besides science. In fact, you will have to. But science can lead you to say, look, there was some amazing designer behind this. Kind of like Fred Hoyle said, it sure looks like some master intelligent tinkered with this. It does.
It does, absolutely.
And here's the next thing on that next slide. A code within a code, we've learned, are inside the cell. Codes inside of a code now. You understand what I'm saying?
Yeah.
I want to give you an illustration. I'm going to read from this book by Stephen Meyer. Wonderful book, Signature in the Cell.
Mm-hmm.
He's talking about this that we found out. To get the picture of how this works, imagine a coded message in a letter sent back across enemy lines during the Revolutionary War. And by the way, General Washington and his troops had figured out a way to do this.
Yeah.
If you read the letter in the standard way, the letter describes problems on the farm, the weather, household challenges, progress the children are making in their studies, all of it clear and accurate information. Yet the letter also manages to encode another message about enemy troop strength, movements, supplies of ammunition, and an impending enemy attack. The soldier who received the message can read it because he possesses a key, a cipher that enables him to identify the location of the second message embedded in the first and then translate it.
Wow.
That's called steganography. Stegano means hidden.
Yeah.
Graphy means writing.
Yep.
Steganography is the hiding of a message inside another message. All right. We know that's taking place inside a cell.
Wow.
We figured that out. And can you imagine now the natural selection acting on random variations, producing a message inside of a message?
No.
I mean,
impossible.
it blows your mind.
Yeah. Yeah.
It blows your mind. I mean, that just dwarfs any explanation that says natural selection did that, in my mind.
Absolutely.
We have discovered that in those other 95%.
Mm-hmm.
And it's a long story of how that works, but it's the truth. So the point is, the more we learn about supposed junk DNA, the more it appears to be designed.
Mm-hmm.
So therefore, I would say, again, as Michael Behe does in his wonderful book, Darwin's Black Box, which, by the way, has come out in a second edition, and the case is harder than ever for the evolutionists. Everywhere we look, from the macroscopic to the macroscopic, things look like they're made or designed.
Mm-hmm.
So isn't the most reasonable answer, well, they really were designed?
Yeah, that's exactly right.
Okay. Well, Dawkins wrote a book called The Blind Watchmaker.
Yeah.
And in that book, he argues that while it looks like it's designed, it is not. Natural causes did it, and it's natural selection, acting on natural variation over millions of years, is sufficient to explain for all that you see that looks designed. And I say, that is ridiculous.
Amen. Amen. And that takes a lot of faith for him to say that, by the way.
Yes, it does.
It does. That takes a lot of faith. Well, this has been fantastic. So let's wrap up here and let's see if we can wrap up some thoughts. The argument for design is one of the best arguments that we have. We can all look around nature. We can all just go outside and see, as one person said, it looks like someone's been tinkering with everything that was made. The amount of information, coding, language, purpose, and DNA screams for a designer, a mind behind it all. The fact that proteins are so essential and where they fall in line and how they're already there before more proteins are being made also screams this could not have evolved over millions or billions of years, that there had to be, I guess, as you even think about like time, space, and matter, a marked beginning. This all had to be, created. There had to be a design, with all of that being put into place.
Let me emphasize right there. Michael Behe says that's irreducible complexity.
Explain that. What does that mean?
He means that you have to have everything there. He uses the example of a mousetrap.
Gotcha.
You have to have a board. You have to have some metal pieces. They have to have a spring on them. They have to be organized. If you're going to catch a mouse, all of that has to be there or the mousetrap doesn't function.
It doesn't work.
In other words, that's a machine or a thing that's complex enough. It all has to be present or it doesn't function.
Mm-hmm.
So it's irreducibly complex. You can't reduce that down to small steps over long periods of time that finally works.
Mm-hmm.
Because in the meantime, nothing worked.
Mm-hmm.
So that's just
Irreducible. Say that.
complexity. It is
Gotcha.
so complex, you cannot work without all of it being working there present.
So that forces a person really to either rethink or, I guess you could say, ignore everything you just laid out. And I love that example with the mousetrap, that you just can't build that over time and expect it to work or to accomplish what it's actually trying to function.
And there are
So
only like that in the human body, for example.
I don't know everyone who listens to my podcast. I know people around the world are listening to it. I do know some who are listening to it and what their beliefs or faith may be. What would you want to share as we begin to wrap up for those who may be seeking, those who may be struggling with doubt, those who are just hearing this for the first time and then saying, what am I supposed to do with all of this? Give us some direction with that, Dr. Payne.
Well, I would just say if you're serious about it, then you're going to take the time and energy to do some investigation sufficient to satisfy you.
Mm.
And I would be happy to share a bibliography of materials.
I love that.
Recommend where to start, because you got to start somewhere.
Right.
And for someone who's not a science math person,
Mm-hmm.
doesn't have that much interest in that, then they need to start with a simpler approach and get their feet wet in the idea. So there's some wonderful materials out there available for someone to dig as deeply as they want for as long as they want.
Gotcha.
And most of that's been written in the last 50, 60 years from a creationist perspective, from a believer in intelligent design. And some of the things I wouldn't recommend.
Like what?
Well, some of the
Oh,
books
in that list. Okay.
Some of the books recommended, written by evolutionists, are so biased and strong, and some are written by creationists that are so biased and I think have the wrong approach. So I have a limit to what I recommend because I think the approach matters.
Gotcha. You've mentioned the term creationist too. Explain that. Is creationist, it may not necessarily be one who believes in Jesus, is that correct? But they believe that there is a creator and that there has to be a mind behind all of this.
And to me, if a person is coming from the background of not believing in God at all, they need to get to the point where they believe that there's something beyond this world that had a part to play in making this world.
Mm-hmm.
Surely that would then stir within a person a desire to learn more about who that is. And is there more to learn about that person? And I believe the next step then is to go to the Bible, which I think you could demonstrate is not a book written by any human.
Mm.
It's got so many features about it.
Mm-hmm.
Convince someone that it's written by deity.
Mm-hmm.
But that's another set of arguments. That's not what we're talking about here.
Yeah, that's right.
I hope that's where it would lead someone to go further investigate. Where else can I go to learn more about God?
Gotcha. Yeah. On the day of this recording, I got a haircut earlier today. I was talking to my barber about my podcast. I Can Do podcast with Benjamin Lee. And he was saying, yeah, I listen to a lot of podcasts, and typically there's a guy, he'll ask a question. He'll ask the same question to his guest. And I was like, oh, yeah, what's that? And he said, he said his question is, have you seen any ghosts lately? And I said, well, I'm not asking that question because I don't believe in ghosts. But I said, that is something interesting, having some kind of like ending thought or ending message or something like that. So I had a author on my podcast previously. Last week I recorded with him, and I'll ask you the same question that I asked him, Dr. Payne. And so my final question to you, Dr. Payne, is what's next for Dr. Buddy Payne?
Well, you and I talked a little bit about one of the things I'm trying to do in this latter stage of my life is study more and more about what we've learned in the last 50 years, because I was involved in a heavy workload that didn't allow for much in-depth study for a long period of time.
Mm-hmm.
When you're president of a college, you don't have much time to read a lot and think deeply because you're tied up with so many things. So it's a real privilege to get to go back and dig more and present material that shows the powerful evidence for God in the natural world. So I think that's my calling for the next however long I'm allowed to live.
Yeah.
So that's where I'm going to put my emphasis.
Good.
And I'm glad to get an opportunity like this. And
Yeah.
another one, as I told you, coming up in the fall, where we're going to do some recording of information and material that I think will be useful, and maybe even write a book. Who knows?
Yeah, I'd love to get that. Yeah, I think that would be a blessing to so many people. Well, there's a lot more that we could talk about. Dr. Payne, I really appreciate you and you using the talents that the Lord has given you to help expand the kingdom and help people to learn more about the true and living God. So thank you so much. Final thought for the audience, and then we'll stop there. Don't hang up though, but final thought for the audience.
Oh, for me?
Yeah, leave us with one final thought.
Okay, I would say if you're searching, and first of all, I would say thank you for listening to this, because I have been involved with people, scientists, who are so close-minded about this subject. It's like I mentioned Dr. Lewontin at Harvard. We're not letting God's foot in the door, so they're not even going to discuss it. They consider it an embarrassment. And there are science departments that would not allow me to speak because they're embarrassed that somebody who's got a science degree would still believe such things. It's like taking you back to the dark ages, many folks think. So I would say thank you for listening and opening your mind to evidence. Dig into that evidence and examine it carefully. The more you dig, the more you're going to see powerful evidence for the existence of a grand designer. And then find out who that is. I'm telling you, the scriptures would tell you he's a God that loves us so much, he gave his only begotten son that we might have a relationship with him. So that's what I want for somebody. But I want to start by being true to the science and letting the science lead to the point. There has to be some designer behind this. And let that lead then to the next step, which is what can I find out about him more? And I would turn to the scriptures. Good. Thank you for the time.
Thank you. Thank you so much. Take care. God bless.